The excretory organs in some crustaceans, crayfish, are internal glands or the green glands. In crayfish, crab filter hemolymph filter into the internal gland from the hemocoel. In crayfish crab, hemolymph pressure from the heart is main driving force for filtration. Marine crustaceans have short nephridial canal and produce urine that is isosmotic to their hemolymph. The nephridial canal is longer in freshwater crustaceans, which allow more surface area for ion transport. In crabs, shrimps, and pillbugs, the excretory organ near the maxillary segments are termed as maxillary glands. Insects have an excretory system made up of the gut and the Malpighian tubules. In insects, excretion involves the active transport of potassium ions into tubules from the surrounding hemolymph. Coaxial glands are most common among the arachnids. Recent evidence suggests that the coaxial glands may also function in the release of the pheromones. Some arachnid species have Malpighian tubules instead of or in addition to the coaxial glands. In some arachnid species, rather than excretion, Malpighian tubules seem to function in silk production. Vertebrates gain water by the absorption from the liquids and solids foods in the intestine. Vertebrates take in solute by the absorption of minerals. Vertebrates lose solute in the sweat, feces, urine, spelling mistake, and EIGA, and gill secretion and as metabolic waste. In vertebrates, the major metabolic waste that must be eliminated are the ammonia, urea, or the uric acid. In vertebrates, the major function of the kidney are the filtration, reabsorption, and the secretion. SECRETION and secretion. Vertebrates have two kidneys that are in the back of the abdominal cavity, NA loga, or either on the side of the aorta. In vertebrates, each kidney has a coat of connective tissue called renal capsule, CAPSULE. The inner portion of the kidney is called medulla, और जो vertebrate का outer portion है या capsule और medulla के दरमियान है, that is known as cortex. The structure and function of vertebrate kidney differ depending on the vertebrate groups and the developmental stages. There are three kinds of vertebrate kidney: the pronephros, mesonephros, and the metanephros. The kidney that appear only briefly in the vertebrate embryo and not all in the mammalian embryos is the pronephros. In tetrapod and other ये tadpoles है POLS and other amphibian larvae, the first osmoregulatory and excretory organ of the embryo is the pronephros. During embryonic development of amniotes or during metanephros in the amphibians, the mesonephros replaces the pronephros. The functioning embryonic kidney of many vertebrates and also adult fishes and the amphibians is the mesonephros. In adult reptiles, birds, and mammals, the mesonephros gives way during embryonic development to the metanephros. The physiological difference between the kidney types of vertebrates are primarily related to number of blood filtering units. अब ये डिफरेंट वर्टिब्रेट्स में क्या डिफरेंस है इनकी किडनीज के अंदर नंबर ऑफ ब्लड फिल्टरिंग यूनिट्स किडनी जैसे जैसे डेवलप होगी उसके अंदर ज़्यादा होते जाएंगे Sharks have rectal glands that secrete a high concentrated sodium chloride solution. To reduce water loss, sharks use two organic molecules, urea and trimethylamine oxide or TMO. In sharks, urea denatures protein and inhibit enzyme, whereas TMO stabilizes proteins and activate enzymes. In shark, urea and TMO together in the proper ratio counteract each other, raise the osmotic pressure, and do not interfere with the enzyme or proteins. This reciprocity is termed as counteracting osmolyte strategy. Because the body fluid of the freshwater fishes are hyperosmotic relative to the freshwater, tend to enter the fishes, causing excessive hydration or bloating. Freshwater fishes absorb salts and water by the active transport across their gills. Body fluids of marine fishes are hypoosmotic with respect to seawater, and the water tend to leave their body, resulting in dehydration. To compensate dehydration, marine fishes drink large quantities of water and they secrete ions through secretory cells in the gills. In marine fishes, channels in the plasma membrane of their kidney relatively transport the multivalent ion out of the extracellular fluid into the nephron. The primitive pronephric kidney found in the embryonic fishes and the amphibians and adults of hagfishes. In amniote embryo, adult fishes and amphibians, the functional kidney is mesonephros. The anterior portion of the opisthonephros function in blood cell formation and secretion of sex hormones. In freshwater fishes, the environmental condition relative to the body fluid is hypoosmotic. In salt water, the environmental concentration relative to body fluid is hyperosmotic. In sharks, the environmental condition relative to the body fluid is the hyperosmotic. In amphibian, the environmental condition concentration relative to the body fluid is the hypoosmotic. In marine reptiles, the environmental concentration relative to the body fluid is the hyperosmotic. In marine mammals, the environmental concentration relative to the body fluids is hyperosmotic. In freshwater fishes, the urine concentration relative to the blood is hypoosmotic. In saltwater fishes, the urine concentration relative to the blood is isoosmotic. In sharks, the urine concentration relative to the blood is isoosmotic. In amphibians, the urine concentration relative to the blood is hypoosmotic. In marine reptiles, the urine concentration relative to the blood is isoosmotic. In marine mammals, the urine concentration relative to the blood is hyperosmotic. In desert mammals, the urine concentration relative to the blood is hyperosmotic. In marine birds, the urine concentration relative to the blood is weakly hyperosmotic. In terrestrial birds, the urine concentration relative to the blood is weakly hyperosmotic. In freshwater fishes, the major nitrogenous waste is ammonia. In saltwater fishes, the nitrogenous waste is ammonia. In sharks, the major nitrogenous waste is ammonia. In amphibian, the major nitrogenous waste is ammonia and the urea. In marine reptiles, the major nitrogenous waste is urea and ammonia. In marine mammals, the major nitrogenous waste is urea. In desert mammals, the major nitrogenous waste is urea. In birds, the major nitrogenous waste is uric acid. Amphibians take up water and ions in their food and drink through the skin and through the urinary bladder. Urinary bladder may be water or ions absorbed. The urinary bladder of the frog, toad, and salamander is an important reservoir of water and ions. In amphibians, when the environment becomes dry, the bladder enlarges for storing more urine. If the amphibian becomes dehydrated, a brain hormone causes water to leave the bladder and enter the body fluids. Ectotherms generally obtain heat from the environment, whereas endotherms generate their own body heat from the metabolic processes. The temperature regulating center that functions as a thermostat with a fixed point is called the hypothalamus. The functional unit of the kidney is the nephron. In most reptiles, kidneys are the primary regulatory organs for controlling the water and osmotic balance of the body fluids. A major site of water loss in the mammal is the lungs. Some deserts and the marine reptiles and birds built up the high salt concentration in their bodies to get themselves of excess water. These animals have salt glands. To reduce evaporative loss from the lungs, many mammals have nasal cavities. The filtration device of the metanephric kidney consists of over 1 million individual filtration, secretion, and absorption structures called the nephrons. At the beginning of the nephron, there is a filtration apparatus called glomerular capsule. In each glomerular capsule of the metanephric kidney, an afferent arteriole enters and branches into a fine network of capillaries called the glomerulus.